How does a cell know when it needs to make a product like a protein or an enzyme? It's kind of like supply and demand in the business world. If Best Buy has a warehouse full of Xboxes, it's not going to order more Xboxes because it can't even sell the ones that it has. So it's kind of like that supply and demand. It's the same type of thing with cells. So this first mechanism, which is called feedback inhibition, this is when excess product basically shuts off the pathway of making more product. And this makes sense because why should the cell spend a lot of time, energy, and resources into making something that it doesn't need? And this is when that end product actually works as an inhibitor. If you take a look at this pathway here, there's an enzymatic pathway where we have several enzymes and different intermediates to make the end product threonine, which is an amino acid. So if the cell has a lot of threonine, does it need to make more threonine? No, because it would be wasteful. And it could also build up to toxic levels, which would not be a good thing. So in this case, threonine acts as an enzyme inhibitor. So you can see where that threonine actually binds to that first enzyme in sequence, and it kind of denatures that enzyme so that it can't bind to its substrate. And it shuts off that whole process of making more threonine. So this is a case of that supply and demand that we could see in both prokaryotic and eukaryotic cells. A mechanism of gene regulation that we really see in only prokaryotic cells, maybe a few eukaryotic exceptions, are operons. And operons are a group of closely related genes, and this is how enzyme and protein production can be regulated. So on the DNA, if you remember with transcription, there is a promoter region, and that promoter region is where RNA polymerase binds. Next to that promoter region is an operator region. This is a site on the DNA where something called a repressor protein can bind. When that repressor protein is bound to the DNA, it blocks transcription. It's almost like putting a giant box on a train track, and the train is not going to be able to move down past that big box if it encounters it. So this first operon that we are going to talk about is enzyme induction. So the enzyme that is needed to make a product is only going to be made when there's very little product available. So this is the lac operon. So if you take a look at the top picture, and I know there's a lot of things going on in this picture, it's very busy. If you take a look at the top picture, you see a strand of DNA. And on that DNA, you see a promoter region, and that's where RNA polymerase binds. Next to the promoter region is the operator region of the DNA. And you see that blue kind of kidney bean shaped thing? That's the repressor protein. So when that repressor protein is bound to the operator, that's like that big box on the train track. You can see that pink RNA polymerase binds to the promoter region, but because that repressor protein is bound to the operator, it's blocked. If you see past that operator region, there's these purple genes, Z gene, Y gene, and A gene. Those need to be transcribed in order to make an enzyme. And the enzyme in this case is lactase. And lactase is an enzyme that breaks down lactose. So lactose is a disaccharide. It is made up of two sugars, glucose and galactose. And when lactose is there, that means that the bacteria want to break it down. They like glucose. Glucose is kind of like chocolate for them. So they want to break down that lactose so they can get at that chocolate. So if you take a look at that first picture on the top, it says that lactose is absent. So if there is no lactose, would the cell need to make lactase, that enzyme, to break it down? No, it would not, right? Because why bother making that enzyme if there's no lactose for it to break down? So in this case, the repressor protein is bound to the operator, and it is blocking that train track. So when RNA polymerase binds to the promoter region, it's blocked and transcription can't happen. If we take a look at the bottom picture, this is when lactose is present. So if that sugar is present, does that bacteria need to make that lactase to break it down? Yes, it does, because it wants to get at that lactose, break it down to get that glucose. So you can see on the DNA that we've got our promoter region, and RNA polymerase binds to the promoter region, but you notice that the operator is free and clear. There's no repressor protein. If you take a look at where that repressor protein is, so look at that blue kind of kidney bean shaped, lactose is bound to it. So when lactose is present, it binds to the repressor and basically pulls it off of that operator. Now those genes are free and clear for transcription to happen. And this is a good thing because now that cell can go through transcription, translation, and then get that lactase to break down that lactose. So that is an example of one operon. There is an example of another operon, which is the TRP operon, and this demonstrates enzyme repression. This blocks transcription of genes that are currently unnecessary to the cell. So we're going to look at another picture here. So this is the TRP operon. So if you take a look at that top picture, and this is a little more simplified than the LAC operon, but you can see the DNA. So you can see the promoter region. Remember, that's where RNA polymerase would bind. Next to it is the operator region. That's the one in yellow. And then you'll see the genes beyond it that can be transcribed to make the end product, tryptophan, which is an amino acid. So when there is no tryptophan, that cell is going to need to make more. So you notice there's no repressor protein bound to the operator, so transcription can happen. On the bottom picture, when tryptophan is present, there's no need to make tryptophan because the cell has enough. So in this case, the tryptophan is activating the repressor. And there you can see the tryptophan bound to the repressor and activating it so that it's bound to the operator region. So in this case, we're activating that repressor. With the LAC operon, we're actually pulling that repressor off. In either case, it has to do with that supply and demand. So as I'm thinking about the operons, I like to ask myself a series of questions. So when does the cell need that product made? So in the case of the LAC operon, remember those genes are going to transcribe to get that enzyme lactase, which is needed to break down lactose, that disaccharide. So if we go through and we ask ourselves a series of questions, if lactose levels are high, does the cell need lactase? Yes, it does, because lactose is available. That bacteria is going to want to break it down so that it can get that monosaccharide, that glucose. So in this case, where is the repressor? That repressor is going to be bound to the lactose. So remember that lactose, that presence, is going to pull that repressor off the operator. Can transcription happen? Yes, because that repressor is not bound to the operator and DNA is not blocked. Will lactase be made? Yes, it's not blocked. Transcription, translation will happen. So if we do the flip side, if lactose levels are low, does the cell need lactase? No, it doesn't, because there's no lactose to break down. Why bother making those enzymes if they're not needed? So in this case, where's the repressor? It's bound to the operator. Remember, there's no lactose available to pull it off. So that repressor protein, that little blue kidney bean, is going to be bound to the operator. So can transcription happen? No, because that's that big box on that train track. DNA is going to be blocked. So when RNA polymerase binds to that promoter region, it can't get past that repressor protein bound to the operator. So will lactase be made? No, lactase won't be made. We can do the same thing with the trip operon. So if tryptophan levels are high, does the cell need to make more tryptophan? No, it doesn't, because it has enough. So in this case, where would the repressor be bound? The repressor would be bound to the operator, right? Can transcription happen? No, because DNA is blocked. Remember, in this case, that high level of tryptophan is going to activate that repressor so that now that repressor is going to be bound to the operator. So because it's bound to the operator, will tryptophan be made? No, it won't. If we take a look at the flip side, if tryptophan levels are low, does the cell need tryptophan? Yes, it needs to make more. That tryptophan, that amino acid, is going to be important in making its proteins and enzymes. So in this case, where is the repressor? It's inactivated, because remember, we needed that tryptophan to activate that repressor to be bound to the operator. So can transcription happen? Yes, because that repressor protein is not bound to the operator, the DNA is not blocked, so transcription can happen. So ultimately, will tryptophan be made? Yes, it will, because DNA won't be blocked. So I know that this can be kind of abstract, thinking about these amino acids and these enzymes. Oftentimes what I like to do is think about something that I can relate to. So if you think about something in the real world, so if you are making something in the kitchen, so I love guacamole and chips. So I think about, okay, if I have chips, do I need to make guacamole? Yes, because I want something to eat with my chips. If I don't have chips, do I need to make guacamole? No, because I don't have anything to eat that guacamole with. So whatever you can relate to as sort of a supply and demand, if you think about things that you do in your daily life, if you think about things that happen in the business world, that can help you as well. Or if you can wrap your head around the tryptophan and lactose, then you can kind of go through those scenarios and practice drawing out the DNA. You can draw out the DNA and think about those different scenarios. Will transcription happen? Won't transcription happen? And these are ways that prokaryotic cells can regulate when they make certain products.